Okay, so we're here once again thanks to another snow day. So this is the video lecture for Internal Transport 2. So we're going to talk about how materials are transported to specific locations within unicellular organisms. And then we'll explain how differences in water potential drives water movement in plants, and how active transport, osmosis, and differences in pressure contribute to movement of sugars in phloem tissue. So we're focusing on the internal transport in unicellular organisms and plants in this lecture. So we'll start with the first objective, talking about unicellular organisms. We'll do this in class. So you should recall this table from your book, and if you did your reading guide, you'll remember this. So there are three main cytoskeletal elements within cells. Actin filaments, which are basically these strings of globular proteins that are, there's two strings that are twisted together, two coiled strands. And among other things, they maintain cell shape by resisting the tension or the pull happening on them. They also help to divide animal cells in two, but we're not talking about animal cells today. But they do move organelles and cytoplasm in plants, fungi, and animal cells as well. There are also intermediate filaments. So these are fibers that are wound together to form these sort of cable-like structures. And similar to actin filaments, they maintain cell shape by resisting tension or pull on them. And then microtubules are like actin filaments, they're globular proteins, but in this case, it's strings of them that are coiled to form these tubes. And unlike actin filaments and intermediate filaments, these structures, these microtubules, are doing the opposite. So they maintain cell shape by resisting compression as opposed to resisting tension. These are the structures that you find in the cilia and flagella of many animal cells, animal cells, but we're not talking about those right now. They help to move chromosomes during cell division, and they provide tracks for intercellular transport. So you're going to see all three of these cytoskeletal elements in our discussion. So when we're talking about transport within unicellular organisms, there's the cytoskeletal elements that work together to move things through the cell. This is a picture of organelles within a cell and some of the cytoskeletal elements. So the real question is, how does the cell know when, say, proteins or a variety of compounds and molecules are in the cell or are being processed in the cell? How do they know where they need to go? Well, they do know, sort of. So if we take the example from the nucleus, so a typical cell imports or exports over 500 molecules through each of 2,000 to 5,000 nuclear pores every second. So if you want to put that in some sort of context, your average machine gun is firing about 1,000 rounds per minute. So this is 500 molecules per second. Okay, so it's fast. So it's transporting a lot of material. And here I'm showing you a slide, cross-sectional view through a nuclear envelope. And here's, a pore cell right here. And here's a stylized diagram showing you a pore where there are ribosomes and mRNA on one side, proteins and building blocks and DNA. So things being transported into and out of the nuclear membrane. So proteins that are produced in the cytoplasm need to enter the nucleus. If they do, they frequently have these tags on them that are sometimes referred to as zip codes, but that's a shorthand. And so these zip codes are basically a series of, or a string of amino acids that serve as a signal. And in the case of things that are going, that need to enter the nucleus, they're called the nuclear localization signals, so NLSs. So how does this work? So the NLS marks whatever that protein is for transport through the nuclear pore complex and allows them to enter the nucleus. And so you can think of this as being like having a ticket to ride a train or fly an airplane. If we take another example, say from the endomembrane system, so this is just a stylized diagram that gives an overview of what's happening within the cytosol. So proteins that are found inside organelles are actively imported into the cytosol, and the endomembrane system produces, modifies, and then tags these proteins, and then it literally sorts them according to those tags that they wander off in vesicles, and they're transported to where they need to go. So let me show you kind of overview of how that system works. You'll probably remember this from cell. Most of you should have already had this class. You all should have had this class already. But just to review, so the signal hypothesis works kind of as follows. So remember, all of this starts with the ribosomes that are outside the rough endoplasmic reticulum. So all proteins start to be synthesized in these free ribosomes. And so what we're showing you here in green is that protein. Next to it, though, is this ER signal sequence. So proteins bound for the endomembrane system have a 20 amino acid long sequence. And you can see that in these little globular structures here. And once that's produced, the ER signal sequence binds to a signal recognition particle. So you can see that in green here, this sort of structure here. And then they move towards the rough endoplasmic reticulum and bind to a receptor here. So you see the receptor here, and then the translocon, which is going to actually allow the protein to enter the cell. So once it attaches to this receptor, the conformation changes. That allows the protein to enter into the lumen of the cell, and it's almost like imagining if you're eating a string of pasta and you just suck it in. So it sucks that protein in. The SRP branches off. The signal actually helps to hold it in place while the protein enters the membrane and is fed in. And And the last part is that once it's all the way fed in, then all the components break off, the port closes, and then the protein is removed. And then it goes into its 3D conformation according to its polarity. So once that happens, right, so that's what's happening right here in the top part of this diagram. So then what happens is those proteins are transported into, wrong part. So it starts here, right? It's then transported out of the rough ER in this vesicle and then enters into the Golgi apparatus. Once there, each protein that comes out has a molecular tag. Ooh, where's my cord? There we go. Which you can kind of see. So it's going to go through the endoplasmic reticulum. It's going to come out through a vesicle, and it's going to have a tag on it. And let me show you that. So it enters the ER, exits the ER, protein enters the Golgi apparatus, exits, and it's going to get secreted from the cell. But here, when we're still inside the lumen of the Golgi, so there are all kinds of different proteins that have been produced here. And notice they each have these different tags depending on where they're destined to go ultimately. So within the Golgi, the proteins are sorted according to their tags. And once they have a group of them together, they'll then be enclosed in a vesicle, and then they'll be transported to wherever their destination ultimately is. And once they get to that destination, the vesicle will then open up and then dump its contents. So there is a system for transporting all kinds of materials through a unicellular organism and through our cells as well. So we know where they're going, but then how do they get there? So we said through the vesicles, but how do the vesicles know where to go? How are they, are they just, bubbles that are floating through through the cell just kind of randomly? Not really, turns out. So microtubules are what perform a part of that function. So yes, they do function in separating chromosomes during cell division. We're not really talking about that just now, though. But they do serve as what's sometimes referred to as railroad tracks for vesicle transport. So you can see an electron micrograph here of a microtubule track, and here's a video image of a microtubule track. So here's kind of the lowdown on what happens. So vesicle transport requires a motor protein called a kinesin. And so you can see a micrograph here of a kinesin. And so here's a cartoon of roughly what these things look like. So there's a head region right here that binds to the microtubule, and you can see it wound here into its cylinder. And then there's a tail region here, and this is what attaches to the vesicle and carries it around almost like a balloon on a string. And so what's happening is the kinesin sort of walks kind of like a duck or maybe like a clown with really big shoes on, like they frequently have. So what happens is this is very high energy cost. And so what happens is with each energy expenditure, there's a conformational change, and then the head, the dual head, if you will, of this kinesin then will do this walk down the microtubule and transport it to wherever this vesicle needs to go. So watch this video, and I'll just kind of chill out while you see what happens. So what they're indicating to you here is that there's a lot of these microtubule elements, and there are. This is some seriously dramatic music. All right, here we go. So here's the microtubule forming. Here it's breaking down. I don't really care that you see this part, really. All right, so then here we go. Here's this giant vesicle being transported by this kinesin as it walks like a big duck down the microtubule to where it needs to go. And they're not the only thing. So mitochondria and other organelles will travel along these microtubule railroad tracks. Pretty killer, these videos. I love these representations of what's going on in the cell. No, don't do it again. There we go. Okay, so that's just a brief overview of what's happening within the cell. So now let's talk about internal transport through plants. In particular, let's talk about water and phloem. So recall, and you'll see this in lab this week, so there's two major players when it comes to water transport in plants. So you have, within the xylem, so within your vascular tissue, within the xylem, you have tracheids and you have vessel elements. So this is a stylized diagram and then a real microscope slide showing you what these elements look like. And you should notice some major differences between these two. So tracheids have a tendency to be smaller in diameter. They have really small pores in their ends, and they're end to end, and water does flow through them. In the case of vessel elements, these generally have a larger diameter, have much larger perforations at their ends, and so you can imagine this is going to allow a lot more water to flow through them than you find with tracheids. And what you'll learn in lab is that different plants have different forms of xylem cells. Some have tracheids, some have vessel elements, and some have both. An important note, by the way, is that when these cells develop, when they reach maturity and then begin functioning to transport water, they're dead at that point. And so as a result of that, they function as a support structure as well. Now, we've talked quite a fair bit about various forces of water, their structure, important aspects of water, and then we've also talked about diffusion, osmosis, and then active transport. Now, water cannot be transported actively. It's only through diffusion or through facilitated diffusion. But bear in mind that most of the movement of most of the internal transport that you see in biological organisms is going to be through a membrane, and it's usually through a phosphobilipid, a bilayer, right? And so you're either going to have osmosis, so it's not just going to be free movement for many particles, or it's going to be actively transported across. And in the case of water, you're usually talking about diffusion, or you have pores like aquaporins involved. In this case, what we're talking about is water potentials. So we're talking about the movement of water from the roots, all through the shoots and up to the leaves and beyond. And so we've mentioned water potential before. And so in an ideal world, maybe I shouldn't put it that way, under general circumstances, so not extreme circumstances, you're expecting that there's going to be a fair amount of water in the soil. So there's going to be high water potential. And then as you move up through the roots, through the trunk in the case of a tree, and up to the leaves, water potential is going to go down. So the further up you go, the lower the water potential is going to be. There can be some variations on that theme. So if you have a high, a very dry environment or salty soils, then you may have lower water potential than usual in the soil. If you're in a very humid environment or in the tropics, you're going to have a higher water potential at the leaf surface. But generally speaking, water potential is higher at the roots, lower at the leaves. So how does this impact water moving through a plant? So water moves from roots to shoots via three forces. So we're going to talk about root pressure, capillary action, and then the cohesion tension hypothesis. So just to refresh you on those tissues, so if you're taking a cross section, let's say through a root, recall that there's several different layers that water is going to have to move through just into the root to get to the xylem, and then it's going to have to work its way from the xylem up to wherever it's going to go. So what you have then is you have root hairs on the outside of the epidermal layer. You then have several cells within the cortex that water will have to transport through. You then have the endodermis, which is indicated here in yellow. You have the pericycle, so you have cells within the pericycle. And then you have in the case of the root, these red xylem cells, and you have a little bit in kind of this greeny, bluey color. That's supposed to be phloem. So don't forget that even roots need energy. So at some point, phloem has to bring nutrients down to it to use. So water is going to have to navigate its way from the root hair over to the xylem. So how does it do that? So it's going to do this along a water potential gradient. And let me show you how that works. So it's going to travel through along this blue trail through three possible routes. So this is a figure from your book. So the three roots are the symplastic root, the transmembrane root, and the apoplastic root. So let me go through this briefly. So the symplastic root is probably far and away the fastest route. So once the water enters the cell, however it enters the cell and the root hairs, it can travel along these plasmodesmata. So recall that plasmodesmata are these gaps between the cells. And when you have these gaps between the cells, it's still lined by the cell membrane, but it's as if the cell membrane is one and the same between the two cells. Okay, so they're joined together. So this is called a symplast. So water can then travel, once it's in the cell, it can travel along these plasmodesmata until it eventually, so it's going to go through the cortex until it eventually reaches the endodermis. So this yellow right here, this is the Casparian strip of the endodermis. So let me back up a second. So here you go. Whoop. So root hair, epidermis, through the cortex until you reach the endodermis. So this is the endodermis, right? And this yellow part is the Casparian strip. And I'll talk about this in just a minute. So going back to the water. So symplastic root through the plasmodesmata. The transmembrane route is through water channels. So this can be through aquaporins, or it could diffuse passively through the cell wall. But just remember that when it does that, it's going to be a much slower route than if it went through the aquaporin or through the symplast. But then once it's into the cell, it can travel through the cell and then through the membrane, through the cell, through the membrane until it reaches the endodermis. The third route is the apoplastic route. So it can, in fact, enter the cell wall itself, move through the cell wall until it eventually reaches the endodermis. Now, whether it reaches, whichever way it goes, whichever of these three ways it goes, once it reaches the Casparian strip, there's going to be a bit of a problem. So within the Casparian strip, this yellow strip here, this is a waxy layer that blocks water. The water is going to have to find some way to get past this Casparian strip. Now, if it goes through the symplast route, there's actually an opening in the cell right here, part of the plasmodesmata, that it can then pass through to get to the xylem. So that would be the easiest way. In the transmembrane route, it could go through the cell, but it's going to have difficulty getting past this Casparian strip. And same thing with the apoplastic route. It's going to have trouble getting past this Casparian strip. So what's going to have to happen then? Oh, sorry, just to review. The Casparian strip is a thick layer of waxy suberin that blocks the apoplastic pathway at the endodermis. Okay? So in order for it to get past this, water and solutes must get into the cytoplasm at some point. So it can travel along this cell wall, but once it gets here, it's going to have to get into the cytoplasm of the endodermal wall and then pass through, either through the cell wall itself passively or through the plasmodesmatal opening. So as water and ions are traveling through these cells, they can only enter the xylem after passing through two membranes. So they have to enter the symplast, and then they'll have to leave the symplast. Oh yeah, and specific channel carrier proteins control what moves into the xylem, and it also blocks certain things from getting into the xylem as well. So it blocks heavy metals, among other things. Okay, so that's how water would travel once it's in the root. That's how it would travel along the root hair and then ultimately end up in the xylem. But why does water move into the root? And we've talked a little bit about this already, but just to review, roots accumulate ions from the soil, which lowers the water potential. And when that happens, ions are pumped into the xylem, drawing in water from nearby cells, and positive pressure forces the water up the xylem. So let me show you a little bit in a little bit more detail kind of how that works. So roots accumulate ions from the soil, lowering water potential. Ions, so meaning that water is going to be pulled in to the root. Ions are pumped into the xylem, and when that happens, they frequently bring water with them into the nearby cells. And then positive pressure can force water up the xylem. But there's a little bit of a limit to that, right? So water can only go so far up a plant. So stomata close at night in most plants, not in all plants, but in most plants. And that minimizes water loss and slows transpiration. In low-growing plants, root pressure can force water droplets out of the leaf margins, and that's what you're seeing here. So if they're really short plants, then while those stomata are closed at night, water is being drawn into the roots because of that solute water potential gradient. And then when it comes into the root, then if they're short enough, it can actually force its way via capillary action. It can force its way so far into the plant that it actually starts to come out the ends of the tips of the leaves. And when that happens, that's called guttation. So just to review capillary action, and you'll do an experiment in lab, and I'm sure these are forces that you're already familiar with, but recall that water has some really interesting properties about it. So it exhibits cohesion, so the water molecules are tightly bonded to each other. They exhibit adhesion, so it's frequently attracted to the sides of a vessel if it's in a vessel. So in this case, they're showing you a glass capillary tube, but it's also true for xylem cells. And then as a result of cohesion in those tight hydrogen bonds, at the surface where it encounters the air, it creates what's called surface tension. So it pulls the water column up to minimize that air-water interface. And so when you see like droplets on a cell, or sorry, if you put droplets of water on a surface of any kind, as you keep adding each droplet, you see that it kind of curves over instead of flattening out. And that's part of that surface tension and pulling of the water column up. So because of these three properties, surface tension, adhesion, and cohesion, if you put a capillary tube in a vessel of water, like a petri dish here, water will be drawn up the capillary tube. But it's only going to go so far. So there's certain physical forces that prevent it from going very high up. Pardon me. So capillary action, so root pressure and capillary action aren't enough to overcome gravity and push the water all the way to the column of the tree. So how does a tree in particular, any plant, but a tree in particular, how does it get water all the way up to the tops of the trees and to the leaves at the top? Well, in looking at this diagram, so again, we're kind of focusing on trees here. So water is going to enter the roots here. It's going to transport, it's going to be transported to the xylem. It's going to flow up the stem, up to a certain point anyway. And so most people think that the way that it continues all the way up to the leaves is just via capillary action, but that's not true. But then some people will tell you that it's transpiration. So if you look at this cross section through a leaf, it's definitely the case that if you look closely at xylem, water can be not only evaporate within the leaf itself, which would draw more of that water up through the capillary or through the xylem tissues. But of course, by opening up the stomata, you have active transpiration, so water vapor is leaving. And so as it's evaporating, more water could be pulled up. But that's actually not enough to counteract the forces of gravity. And there's some other forces that are actually acting on the plant as well that would make that impossible to account for drawing water all the way up to the tops of trees. So I'm going to run through this in these slides. So water vapor diffuses out of the leaf. It evaporates within the leaf. So again, both are forces that would then pull water out of the xylem. Water is going to move by osmosis into the xylem as a result of active transport of ions into the xylem. So it's going to get in that way too. And then water will enter the roots from the soil as a result of active transport of ions. Active transport of ions. Okay, but none of this, most of this actually is not a result of expending any energy by the plant, right? So this is all driven by the sun, which is driving the evaporation from the leaf. So water in a column of xylem cells moves by bulk flow. So this is mass movement along it's just a pressure gradient. So it's just a simple pressure gradient. So the negative force or pull generated at the air-water intersurface is transmitted from leaf surface to root. Now, in order to do that, that's going to require a continuous column of water throughout the plant. So if you recall, I showed you those tracheids and vessel elements. So those cells are one on top of another, and they're full of water going all the way up from the roots to the leaves. By the way, this is a diagram that shows you some of this. So the xylem is indicated by the blue cells. The purple cells are phloem. So they're rigid and they add structural support. So lignified secondary cell walls allow the tracheids and vessel elements to withstand extreme negative pressure. So what's happening is as you go up the tree, there's going to be an increasingly more and more negative pressure on the cells and on the water going up. We'll do this in class, so skip that for a moment. So let me show you a video that might help to further explain cohesion tension theory and how water can transport up through a tree. Pretty cool, eh? It is cool. It's super cool. Whoop. It occurs to me now that you might not be able to hear this because I'm hearing this through my headphones, so I may have to send you the link to see this. But I guarantee you, I've been going to tell you again and again, plants can do amazing, amazing things. So this is yet another example of the crazy things that plants can do that animals couldn't even dream of. Okay, but let's carry on to objective 3. So that's all we're going to talk about in terms of that's all we're going to talk about in terms of water transport in plants. So now let's talk about transportation of sugars and hormones and nutrients through phloem. So let's talk about phloem transport. So translocation is the movement of sugars through a plant by bulk flow. So remember, that's just a pure pressure gradient. So bulk flow from sources to sinks through the phloem. So a source is tissue where the sugar enters the phloem. Okay, so usually we're talking about places of active photosynthesis, so places like leaves and stems. A sink is a tissue where sugar exits the phloem, and so that's where it's going to be used. So you can imagine flowers and young leaves and meristems and then roots. So sugar concentrations are high in sources and low in sinks, generally speaking, because of course there's active usage of those sugars for whatever those tissues are doing at that moment. Now, the location of sources and sinks in plants can vary depending on the time of the year and the type of plant. So one example would be if you look at things like tuber plants. In summer, leaves are photosynthesizing actively. There's lots of them. They're producing sugars that then get transported into these tubers, which are then stored, the sugar is stored as starch. But say in spring, sucrose is sent to the new shoots. So here they are. New shoots send leaves up, start to photosynthesize, and they actively use this energy source until they've got enough leaves to be photosynthesizing to produce enough sugar to return down to the tubers. So let's talk about the anatomy of phloem. So there's two main elements. So there's sieve tube elements and companion cells. And they're connected to each other by plasmodesmata. So here's your sieve cells, and you can see why they're called that. So these are plant cells that have holes or pores through them, stacked one on top of the other. And then, by the way, I'll get to that in a minute. And then beside them, adjacent to them, are these companion cells. So sieve cells by themselves have few, if any, organelles at all, whereas companion cells do have a sort of cornucopia of organelles. And here you can see the plasmodesmata that allows for movement of particles between the companion cells and the phloem. So sieve tube elements also, among other things, they lack organelles, including nuclei. Companion cells, of course, have nuclei. They have these perforated plates, as I already mentioned, and they have this direct connection. So the companion cells do a number of things, but largely they're supporting the metabolism of the sieve cells or sieve tube elements. So how does it work? Okay, so those are the tissues, but then how does it work with the transport of phloem? So sucrose is moved by active transport from source cells through the companion cells to the sieve tube members. And this is known as phloem loading. So in this diagram, here's your source cell where you have active photosynthesis going on and then the production of sucrose. So that, let's say it's a leaf cell, that's going to be adjacent to a companion cell where sucrose is going to move through, and here's the sieve tube elements of the phloem. And in this diagram, let me just orient you first and then we'll go through the steps. So the sieve tube elements are stacked one on top of the other, and then at some point it's going to move by bulk flow through a companion cell into a sink where that sugar is needed for whatever process is going on at that time. So growth, reproduction, whatever. You should also note that very close to the phloem cells are, of course, the xylem cells that are transporting water, which is going to be needed as part of this process. So recall that water is moving passively. In other words, there's no active transport. There's no energy expended with water. So it's going to flow from nearby xylem into the sieve tube elements, as you see indicated here. And Turner pressure is going to build in these sieve tube elements in the source region, so up in here. So cells in the sink are going to remove sucrose from phloem. Here you go. So it's going to remove sucrose from the phloem by passive or active transport. So both can be occurring here. So just to review again, so sucrose being produced here moves into the companion cells, then into the phloem. And once in the phloem, bulk flow, so just a simple pressure gradient, is what's going to move the phloem through the sieve cells and towards the sink areas, okay? But it then has to pass through active or passive transport through the companion cell and into the sink, okay? So while this is happening, of course, you can imagine that's what's happening is not only is turgor pressure building up in the phloem adjacent to these companion cells, which are adjacent to the source cells, but it's also going to have a higher solute concentration, which is going to draw water from the xylem cells into the phloem, increasing the turgor pressure. And then as the sucrose is transported into these sink areas, the solute level is going to go down, which will then cause water to move back into the xylem cells. So water will diffuse back in. And then the cycle continues. So the net result is high turgor pressure near the source, low turgor pressure near the sink, and that's going to drive phloem moving from the source to the sink by bulk flow. So you'll have this pressure differential. So interestingly, the sugary fluid in phloem is under such high pressure sometimes that aphids, and you're seeing an example here, drinking can't absorb it all, and some is forced out through their digestive system and out their anus. And sometimes ants will come up and suck up this sugary treat for themselves. So win-win. Okay. So there's a one-way flow of sucrose and a continuous loop of water movement. So just to be clear on that. We're going to do that in class. Okay. That's the end of the lecture. I will upload this for you, and then we'll do some of these activities in class. See you on Tuesday, and I'll send more info via Blackboard. Good night.